In the previous video, we have seen that amphipathic lipids used in membranes use two different kind of backbones. One that is using the unit of glycerol, and the second one that uses the unit of sphingosine. We have seen a little bit of what is that called the phospholipids, and now we are going to discuss those that are using either a monosaccharide or an oligosaccharide that are called the glycolipids. In this case, we will have, the glycolipids are using mainly the unit of sphingosine plus one fatty acid. Once this fatty acid has reacted, it's going to be transformed into an amide bond. So we create an amide bond. An amide, which is the reaction between the amino group and the carboxylic acid end of the fatty acid to produce the amide bond. Now, the sugar is going to be attached to the primary alcohol and not the secondary alcohol. That will create again what is called a het. So this is going to be the het. And still we'll have two tails. One tail coming from the sphingosine and one tail coming from the unit of the fatty acid. It is known that many of those lipids, complex lipids, are constantly being synthesized and decomposed in the body. There are several genetic diseases classified as lipid storage diseases, and I am listing some of those diseases or conditions in here. I have listed the Gauchers, Krebs, Fabry's, and Tay-Sachs. Those conditions are by accumulation of some glycolipids. If we look at sphingofospholipids as part of the functions, we will say that they have an important function for nerve cells. The myelin, which is important for the insulation of the nerve axons, and discussing what is the composition of those lipids, it will be 70% lipids and 30% proteins. And one example that I'm presenting for a myelin is when you have a phospholipid with choline. So you have this hydrophilic part and a hydrophobic part. This is abundant in the brain. Now, those that are glycolipids are facing the exterior of the cell. They are going to be part of the membrane, but facing the exterior of the cell. Now, how is that a membrane is going to be made? A membrane is nothing but a bilayer, two layers of phospholipids, also some glycolipids, and units of cholesterol. Let me place one unit of cholesterol. This is called the mosaic. It's called a mosaic because it's not homogeneous. We will have phospholipids that are not attached by any covalent bond. So we say that there are no covalent bonds between any of those tails. So the phospholipids will aggregate by van der Waals interaction, London dispersion forces. But more than anything, it will be by hydrophobic effect. Hydrophobic effect, which has to do with the property of hydrophobic substances of clustering each other to be away from water. Water wants to be with water, so these are going to form clusters. They are not making micelles because having too long hydrocarbon chain will prevent from forming spherical surfaces like in a micelle. Now we will have that in the exterior of the membrane facing out of the cell, we will have the glycolipid part. This is representing the oligosaccharide. None of the glycolipids will be facing the interior or into, that will be into the cytoplasm of the cell. The rationale for that is that Glycolipids are important for cell-to-cell interactions. They are kind of messengers. They are working as if they are messengers. It's giving information from one cell to the other. These membranes are creating barriers, that is, to separate one cell from another, or also to separate organelle. Properties of these membranes are that they are amphipathic because they have a hydrophilic part, so all of the heads that will be phosphate units or amino alcohol parts, they will be making interaction with molecules of water on the outside and also in the interior, because we will have molecules of water in the interior of the cell and molecules of water on the exterior of the cell. And then we will have also some integral protein and peripheral protein that are important for the communication between in and out. Some molecules will pass through channels through the protein, and some ions also will pass by those channels. It will be different kind of transport that I already have discussed a little bit of transport in my iPad video, so you guys please watch those. The last thing that I'm going to discuss before we look at another important part is what is called, another brand new family is called the eicosanoids. The word eicos means 20 carbons, and the family of eicosanoids are all derivatives of one compound that is 20 carbons long, and it has 4 double bonds. All of them are cis. I'm gonna say that they are in delta position number 5, position number 8, 11, and 13. And the name of this is called arachidonic acid. And that's coming on the next video.